So as you can see, I'm not Dr. Janes. Sham and I today are going to be giving the lecture, and it's the first of a two-part lecture on transcription and its regulation. And Dr. Janes is going to finish it. We're going to kind of give you the XYZ 1, 2, 3 mechanism today, the nuts and bolts, and then Dr. Janes is going to go into the regulatory mechanisms more in depth on Tuesday. So the clinical disease wrapper we're going to use for this is a special form of diabetes and as well as anabolic steroids. So diabetes is a group of metabolic disorders characterized by hyperglycemia, so high blood glucose levels, and it's caused by defects in the secretion of the endocrine hormone insulin, the action of insulin, or a very complicated combination of both. So it's a little awkward talking about this as a metabolic disorder, considering you guys haven't done metabolism yet, but you guys are going to have a whole host of lectures on that. I think it's in quarter 4, but just kind of like the main concept you're going to need to understand today is glucose is basically the precursor molecule that metabolism, aerobic and anaerobic, so oxygenated and deoxygenated, are both going to need to start with for your cells to produce energy and function. And if your cells can't get this glucose, they can't make this energy, and they have to turn to alternate pathways, which is when you can get all of these negative side effects. So does anyone know what insulin does in the body and why the dysfunction of it in diabetes is so causes so many symptoms? Any guesses? No? Okay, so insulin, what it does in the body, so glucose is a polar hydrophilic molecule. It's too big to diffuse right through the cell membrane, the plasma membrane, so it requires dedicated transporters to bring it in through facilitated diffusion like we learned about previously. So these transporters must be mobilized to the cell membrane surface in order to do this. So in order to do this, insulin is that molecule that regulates the mobilization of these transporters and where this happens in your body for cells to uptake glucose to be able to produce energy. So chronically high blood glucose levels, if sustained and left untreated, causes a whole host of problems with different organs, and mostly involving fine vasculature, such as the eyes and the kidneys that are particularly sensitive to this high blood glucose, and also there are neural and cardiac implications. So some symptoms here, polyuria, that's frequent urination, so going to the bathroom a lot. If you think back to the G proteins cholera lecture when we talked about the sodium glucose concentrations in your cells and dehydration as a result of the malfunctioning of the CFTR in the gut epithelial cells. High blood glucose levels, once filtered by the kidneys, are going to cause water to be driven out of your cells, and your cells are going to get dehydrated and you're going to have to go to the bathroom because of this persistent glucose in your blood. Kind of correlated to that, polydyspia, so frequent thirst because you're going to the bathroom so much and losing all your water. And interestingly, diabetes is kind of, you can think of it as chronic cell starvation, where the issue isn't the fact that you don't have the nutrients, the glucose is in your body, but it's just staying in your bloodstream and not actually getting absorbed into your cells to be able to be utilized. And then, as I talked about, if you can't get glucose into your cells to start your metabolic pathways to produce energy, your cells are going to have to turn to alternate means. So ketoacidosis is one of these means where you turn to the breakdown of fats. So when you do that, you break fats down into keto acids, which get broken down further into ketone bodies. And these ketone bodies can actually alter the pH of your blood, and you can actually smell this on people's breaths, which is kind of interesting. And some long-term symptoms sustained over your lifetime, so retinopathy, problems with the retina, your eye, diabetic neuropathies, so that's problems with your peripheral nervous system and circulation as your tactile senses are impaired. So if you have a wound, it can be very dangerous because if you have bad blood circulation, your cells can't get to the wound and recognize it to heal it and start the clotting process. You also are at an increased likelihood for cardiovascular and cerebrovascular diseases, so brain flow associated diseases. And there's other effects such as lipoprotein metabolism, which is your handling of fats, as fats and cholesterol are abnormally persistent in the body. And there's also some chronic oral inflammation. So prevalence and economic impact, this is a very common disease. I'm sure you've all have heard of it before. These stats are a little old, but you get the rough order of magnitude impact here. It's interesting just to note that this is kind of known as a disease of developed nations. Can anyone guess why? Yeah? Exactly, yeah. So there's two types of diabetes, which we're about to talk about, and one of them is kind of nutrition-based, so wealthier nations that can afford to eat not as healthy or eat surplus food have a higher prevalence of this disease. And there's also a genetic predisposition of certain cultures and ethnicities. So there's two major types of diabetes. Type 1 is the one that you're born with. It's known as juvenile or inherent, and it's caused by the destruction of beta cells in the pancreas that produce insulin. And this is actually an autoimmune associated disease. So we just finished talking about the immune system. This is a great example of autoimmunity where the autoantigen is insulin itself. And there's a cross-reactivity here as beta cells are attacked and the production of insulin is impacted. So type 1 is actually proportionally rare compared to the second type, type 2. So it's the much more prevalent category, much more common. It's sometimes referred to as mature onset because it results later in life due to different factors. And this relates back to the disease of developing nations, your nutrition, because if you're eating all of the sugar all the time, all of the fast food, your body becomes desensitized to glucose, and your insulin doesn't handle it as well, so you get these elevated high blood glucose levels. And this results from improper regulation of insulin or insulin signaling processes downstream. And the type of diabetes that Dr. James is going to focus on next lecture is a special type called mature onset diabetes of the young, or MODY. And this is sometimes referred to as type 1.5 diabetes. And this results from a defect in transcriptional regulation. So into the nuts and bolts of transcription that you're going to need to understand the mechanism for diabetes. Today, Sham and I are going to focus on the transcription process. I'm specifically going to focus on the process in prokaryotes, and then Sham's going to get into eukaryotes. So kind of like with DNA replication when Dr. James did it, we're going to analyze both pathways, but you're going to see it's going to be a little bit less side by side this lecture. But the general pathway is the same in both. It's just a little bit more complicated in eukaryotes. So I'm going to begin with kind of just an overview and get you some of the terminology you're going to need to be fluent in to understand transcription, and then I'll break it down a little bit further into the individual steps. But the terminology that you're going to need to know, so first up, the transcription unit. So this is the DNA sequence that you want to transcribe to produce protein XY that's going to do whatever function in your cells. Then immediately before the transcription, the DNA sequence is the promoter. And immediately after the transcription unit, the DNA sequence is referred to as the terminator or the termination sequence. And together, the promoter and the terminator set the bounds on your transcription unit. And your transcription unit in prokaryotes could be multiple genes versus in eukaryotes, it's a single gene. And then we have this coding strand and template strand to reference your two DNA strands here. RNA polymerase is going to dock onto the template strand to transcribe the reverse complement, and then the coding strand is the other DNA strand. And this is kind of similar to the parent and the daughter strand we had in DNA replication. So the basic process of transcription, there are 3 major phases if you want to break it down. It's going to be initiation, elongation, and then termination. So the first step in initiation, you're going to have RNA polymerase is going to dock on to your promoter region at the start of your transcription unit. And this binding is going to be aided by sigma factor. And then it's going to open up your 2 DNA strands and dock on to the template strand. And then it's going to transcribe off of the template strand to make RNA 5' to 3'. So the template strand is your DNA strand that runs 3' to 5'. So if you're synthesizing the transcript, which is the reverse complement, it's going in the 5' to 3'. And then as it does this, it's elongating using ribonucleoside triphosphates. So RNA, obviously, compared to DNA replication where we had deoxy, now we just have the ribonucleosides triphosphates here. And your coding strand of DNA is actually going to identically match the base pairs of the RNA transcript you're synthesizing, except for the fact that all of the T's on the DNA are going to be replaced with U's. So this makes sense if you think about it, because your template strand is the reverse complement of your RNA transcript. And then if you have 2 strands that are both anti-parallel, both the reverse complement to this template strand, they're going to be complementary to each other. So then RNA polymerase is going to keep going and elongate the RNA until it reaches the terminator, and then it falls off and you're left with your 2 products here, which are going to be your RNA transcript and the DNA back together as duplex DNA. So a little bit more on some of this. Bacterial promoters, so this is where RNA polymerase is going to recognize the start point for transcription of a gene. And this is kind of a common idea in prokaryotes and eukaryotes, that you want to separate your 2 DNA strands and transcribe off of only that 1 strand, which is your template strand, the 3' to 5' DNA strand. And then you're going to base pair to make the reverse complement. And the only difference between the coding strand and your RNA transcript is the replacement of T's with U's. So again, like I said, the 2 strands, if you have 2 strands that are both anti-parallel to the template strand, they'll be parallel to each other. Does that make sense? Do you want me to try and explain that another way? Is everyone good? Does anyone have any questions too? I should pause a little bit more. Yes?
So does the polymerase copy off the template strand, finish that, unattach, turn around, and then make the other side?
No.
It's just the 1 strand?
It's just the 1 strand, yes. That was a good question. Okay, so then also important to note is with your polarities here, your promoter, your terminator, and any other terminology we use is all going to be referenced off of your coding strand, because that's what matches your RNA transcript that you're making. So therefore, your promoter is upstream in the 5' direction on the coding strand. And this promoter, like I said, is where RNA polymerase is going to bind onto the DNA template and open it up to begin the transcription process. So just like in DNA replication, we have these recurrent features of DNA called consensus sequences. So I just want to highlight here that consensus sequences does not refer exclusively to origins of replication in DNA replication. It simply just means a shared stretch of DNA that is used for a common biological function, and that function can be serving as an origin of replication, or in this case, as a promoter sequence. So once RNA polymerase in prokaryotes recognizes this promoter region and docks on and gets stabilized, it's basically off to the races and transcription is going to happen, elongation, and you'll get your RNA transcript. In eukaryotes, the mechanism of regulation that Dr. Jane's going to get into next lecture is a little bit more complicated, and it can be hindered. In prokaryotes, it's really just a matter of binding to that site after it recognizes it, and as long as there's no other molecule in its way, it's going to go and transcription is going to happen. So transcription begins, just one more thing with the numbering sequence here. Transcription begins at the start point here, so at the very end of your promoter. And the start point is, again, referenced off of your coding strand. It's base pair +1. And then your promoter sequence is approximately 35 base pairs long. And since you go upstream on the coding strand from start point 1, that would make the promoter start at about -35. And then it increases after your start point in the 3' direction, so you'll have all increasing positive in this direction on your RNA transcript. Did I see a hand? Any questions before I move on? Okay. Okay. So a little bit more in depth on the 3 stages. So initiation, once RNA polymerase binds and docks onto the template strand, it's going to briefly stall as it opens up the 2 strands, and then it begins to incorporate nucleoside triphosphates, or NTPs, which are complementary to the template strand. And then these base pairs are going to be added one at a time until approximately 9 base pairs are reached. And then we transition into the second phase, elongation, and that occurs. The reason that there's this kind of stall lag here with 9 base pairs being added is because RNA polymerase, after it binds, it needs to get stabilized and stabilize the opening of the DNA locally before it can kind of be on its way a little bit more smoothly. And it's very important to note here that the entire process of this transcription can be done by RNA polymerase by itself. Whereas in DNA replication, we had the need for helicase and gyrase as we were unwinding, and we needed to unzip the DNA and then deal with supercoiling. RNA polymerase doesn't require any other enzymes to really do this. There are some accessory proteins that can help it bind on and get stabilized, or especially in termination, unbind. But the major transcription is all done by RNA polymerase itself. And then elongation, so that's really the major, this is kind of like the major mechanism of the nitty-gritty actually making the RNA and adding the base pairs on. So this is where you see most of the functions of RNA polymerase, so it can unwind the DNA and stabilize this local opening itself. And then it can add on NTPs as it transcribes. And again, remember it's transcribing. The transcript is being produced in the 5' to 3' direction. And it's going to add about 12 base pairs as it's catalyzing this versus this is a little bit higher rate than the 9 we saw in initiation. And in contrast to DNA replication, where we had the DNA winding and winding on itself, and we got the supercoiling problem that we needed the assistance of gyrase for. RNA polymerase, because it's only opening the DNA locally, it does not have this supercoiling issue to deal with. And the DNA actually rewinds itself back behind it, as you can see here. So again, it's going to elongate RNA 5' to 3', moving along the template strand in the 3' to 5' direction, which is anti-parallel. So it's very easy to get turned around in all these polarities between the different pathways. So take a second and think about that. Yes?
So are you saying it doesn't have to do with supercoiling and one replication model?
Yeah, it doesn't have to deal with the supercoiling problem because it's only opening the DNA very locally versus in DNA in DNA replication, you had the whole replication fork, and you had the you were synthesizing off of the 2 strands. Here, since we're only synthesizing off of the 1 strand, it's a much more local opening.
So when you say 12 base pairs at a time, so does that mean it's like opening up this section, like transcripting whatever the 12, and then jumping to the next section, like so it's stacks of 12?
It's not. It's going smoothly. It's just that at any one time, there's 12 base pairs kind of being docked within RNA polymerase and being about to be added. Any other? Yes?
What's the importance of the coding strand?
The importance of the coding strand is the fact that we reference everything to it and the fact that it does identically match your transcript.
So is it looking at both the coding and the template strand?
No, RNA polymerase is docked onto exclusively the template strand.
Yeah, I'll just add to that, like, the template is just serving as, like, its template. I wish it was named something different, but the coding strand is what actually has that information for the gene, and as Emily mentioned, all the promoters that make the RNA polymerase go there.
Okay. So then lastly, how does RNA polymerase know when to stop? So this occurs, RNA polymerase is going to keep elongating until it hits the 3' end of the transcript. So how this is done here is actually through a secondary structure that gets recognized. So RNA polymerase, as it's elongating, it's going to transcribe a section that is self-complementary to a section immediately after it. So what that means is that the regions, since they're self-complementary, they're the reverse complement of each other, if they're close enough together locally, they can actually kind of wrap back on itself and bind together with hydrogen bonding. And so when this happens, this hairpin loop structure is formed here. And it's stabilized by hydrogen bonds here between paired ribonucleotides. And to do this, you need a stretch that's long enough, and they need to be held in close proximity. So again, it's going to be GC rich because you have the 3 hydrogen bonds per base pair pairing versus the 2 with when you have A and U. So the creation of this secondary structure is actually what is recognized by either RNA polymerase or by accessory proteins to actually tell RNA polymerase to unbind and stop the transcription. So secondary structure with reference to proteins, we talked about alpha helices and beta sheets. Here, this refers to exclusively this hairpin structure and the ability of the RNA transcript to actually wrap back and bind to itself. So the hairpin formation, as you can imagine here, it's not instantaneous, the recognition of this. There's some lag between RNA polymerase or those accessory proteins recognizing the hairpin structure as it's formed. So as a result, your RNA transcript is going to be a little bit long, and you're going to have it trimmed later on, as well as this actual hairpin is not kept in the RNA transcript. It gets cut off as well. Okay. Oh, yes.
Is there a name for like the sequence that the RNA will read from the hairpin? Is that like the terminator, or is that something else?
It is, yes, the termination sequence is what is referenced. Do you know, is the termination sequence the actual, is it after or is it before the hairpin?
I think it's, I'm not sure where it is, but it's the actual structure, not just the like base pairs.
Yeah, but I believe that the hairpin structure, it can be formed by a number of different sequences. There's not just, it's not like a promoter where there's kind of like preserved one. It's any 2 self-complementary sequences. Okay. Yeah.
AB switch.
Do I have the other one on still?
Go. Good job.
Okay.
Okay, so does that also go to the end of the lecture hall? Okay. So I'm going to pick up from what Emily built on to talk about eukaryotic transcription, which has all the features that we kind of talked about, but more complicated and has a lot of elaborations. So the first difference you'll notice in prokaryotic and eukaryotic transcription is that there are 3 RNA polymerases, as opposed to just the one that we talked about just now. We'll be focusing the most on RNA Pol II, which makes pre-mRNA, so the precursor to the messenger RNA from which proteins are translated. The other 2 types of polymerase make ribosomal RNA, or rRNA, or transfer RNA, tRNA, which are both involved in the actual translation process. And Dr. James will talk about that in a future lecture on translation. So, and I'll point out here that the before all of these RNAs refers to before splicing. So this is in the first pass through the genome for transcription. It includes both introns and exons. And after the splicing occurs, they become mature rRNA, mRNA, or tRNA. And then one thing that, how does a polymerase know which type of RNA it's about to be making and to go to the right part of the genome? That specificity comes in from promoter sequences. So the 3 promoter sequences for these different types of polymerases are going to be different. And that is a way to confer specificity. So let's talk now about the RNA Pol II promoter. As Emily did a good job laying out before, to orient you again, this is the DNA strand. And now, instead of looking at both strands, we're looking just at that coding strand. So this is one strand of DNA. And this position here at the +1 is that transcription initiation. And around that are these consensus sequences that indicate to a polymerase where to dock and start transcription. So right at this +1 around it is what's called the initiator sequence. The PY in this refers to pyrimidines, so that can be a C base or a T base. It's interchangeable. Along with the initiator sequence is upstream of that is what's called the Tata box. And it's really cleverly named that because it has T's and A's in it. So it's just stretches of T's and A's that are upstream of all of these transcription initiation sites. So together, the initiator sequence and the Tata box make up what's called a core promoter. And this is a core promoter for RNA Pol II and not for the other polymerases that I mentioned. However, as in bacteria, this was sufficient. Just having the promoter sequences in the DNA was enough to recruit a polymerase. In eukaryotes, you need additional factors to confer specificity. And one of those is a transcription factor. So these are proteins that are able to bind both DNA and another protein to start with transcription. So let's talk about the transcription factor that is required for RNA Pol II. So this idea of a transcription factor can be used as an interface between gene sequences and proteins to regulate transcription. And it's a very large family of proteins, and one type of them is called a basal transcription factor. And what makes it basal is that it's required and essential for all transcription involving RNA Pol II. So there are transcription factors that are not basal that might change relative to what gene is being transcribed, but basal transcription factors are always at promoters if a gene needs to be transcribed. The basal transcription factor for RNA Pol II is TF2D, and it binds that Tata box region upstream of the initiator sequence. So the transcription factor 2D needs to be there, but it's not the only signal that's necessary. It starts the formation of a pre-initiation complex that composes of many different proteins. And that pre-initiation complex tells RNA polymerase II to come to that locus and start transcribing that gene. So again, the sequence of events is that TF2D comes in and binds that TATA box. It then recruits other proteins to form the pre-initiation complex. And then RNA Pol II binds that pre-initiation complex and starts transcribing the gene. It's important to note that all of these proteins involved in initiating the process don't move with the polymerase. They stay in that upstream promoted region, and they're just the signal for RNA Pol II to come there and start transcribing. And then so the polymerase moves forward and the gene is transcribed. Any questions on that? Yeah. Yeah, it's like I planted you there. That's a great question. And that's actually the basis of something that's become pretty well established now. Is called transcriptional bursting. So the idea is that the rate limiting step in transcription is really the assembly of this pre-initiation complex. And once all of these factors are there, multiple polymerases tend to come and transcribe the gene over and over again. So one locus gets transcribed a bunch of times at once, and it's called, yeah, transcriptional bursting. Any others? So after mRNA is transcribed from, again, as Emily mentioned, from the 5' to 3' directions, that's still pre-mRNA. And several modifications are made to it before it becomes mature RNA from which proteins are translated. So the 3 modifications that we'll talk more about are 5' capping, so that happens at the 5' end, poly-A tailing, which happens at the 3' end, and after both of those have happened is splicing. You guys might remember splicing from central dogma stuff and how B cells class switched. Same concept, but we'll talk more in detail about how it actually happens. So the 5' cap. On the right here, the first base in green is that first RNA base that was transcribed at the 5' end. At the 5' position of that base, a modification is made by dedicated capping enzymes. This modification adds a 7-methylguanosine to that first base. And it's called a backwards cap because as opposed to all the nucleotide polymerization we've talked about so far that happens in the 5' to 3' direction, this is a 5' to 5' polyphosphate link. So what that does is creates like a very specific chemical feature in that RNA that is structurally different from all the other polymerization so far. And so again, I mentioned these are, the reaction to add the cap is catalyzed by this capping enzyme, and the cap itself, by its unique chemical feature, can then be recognized by cap binding proteins. And these serve a variety of functions. So this cap signal gets flags that pre-mRNA transcript for nuclear export. And why is nuclear export important? Does anyone have a guess? Yeah. Yeah, you're right. So mRNA is made in the nucleus, but translation happens outside the nucleus. And to do that, you have to get mRNA across the nuclear membrane. And this 5' cap is a signal for that. Additionally, once these cap binding proteins bind to the 5' cap, it's kind of a protection for that free end of mRNA. So we talked about exonucleases for DNA. There's also exoribonucleases that can degrade free ends of mRNA. And this helps protect against that. And the last feature of the 5' cap in terms of function is that it is a binding site on the ribosome that performs the translation. So it's particularly important for what's called cap-mediated or cap-dependent translation, which is how a lot of proteins are made in mammalian cells. And so the 5' cap is critical in initiating the translation process there. Another feature of these capping enzymes is that they are associated with RNA pol 2. Any guesses on why it's important, why they need to be associated with pol 2 specifically? Yeah.
Isn't all two, isn't that the one that deals with mRNA?
Yeah. Yeah, correct. So you don't want rRNA or tRNAs to have this cap, so the capping enzymes that added are associated with pol 2. So the second modification I mentioned was polyadenylation, and as Emily mentioned, transcripts are generally longer than the correct gene length, and that's because, again, to reiterate, when you have to make the RNA sequence that forms the secondary hairpin, and then that secondary hairpin has to allosterically modulate the polymerase for it to know that it should stop polymerizing. And all of that has a timeline associated with it. And during that process of allosteric inhibition, the enzyme is still elongating the transcript. So by the time you're done and the enzyme has gone away, you have a too long transcript that needs to be chopped so you have a gene of the correct length. So to do that, there's another consensus sequence towards the 3' end, shown here, that is recognized by another enzyme called poly-A polymerase, or PAP. So that enzyme finds this signal, and that marks the end of the important stuff for this gene that needs to be translated to proteins. PAP will then cut off the excess 10 to 30 nucleotides that might be after that sequence, and then adds a poly-A tail. So that A stands for adenylation, so it's just a series of A bases added at the 3' end of the newly made mRNA. And functionally, this poly-A tail also does similar things like the 5' cap, but on the other end of the transcript. So it protects from degradation. It kind of adds a buffer stretch of nucleotides that protect it from exoribonucleases, and is also necessary for translation, and also provides a signature that allows multiple rounds of translation of the same transcript once it's in a ribosome. Questions about those modifications? Okay. So I'm going to schematize all of this in this cartoon here, and I'll point out what all the things are. So starting with this G, that's the 5' end of this mRNA, and that's the M7G cap. You have this whole mRNA transcript, and the 3' end is here with the poly-A tail in a different green color. And you might notice that this is different from how we've shown mRNA so far. It's circularized instead of linear. So how that happens is that these red proteins, called poly-A binding proteins, or PABPs, can bind that 3' end. They recognize the poly-A tail, and then they circularize the mRNA by binding to each other and binding to the cap binding proteins on the 5' end. So this serves multiple functions as well. One is that when an mRNA is being translated by a ribosome, the ribosome can go around the whole transcript and then just pop back over to the 5' end and go again, instead of having to diffuse back to if it was a linear strand of mRNA. The second purpose they serve is also protective. So you've taken the two weakest parts of a transcript, the free ends, and brought them together. And when they're protected by proteins and brought together, it also further protects them from degrading enzymes, which are shown here. The pink is one of them. It's called a decapping protein. It takes off the 5' cap. And XRN, which is an exoribonuclease, again, chops up RNA. So the cartoon of that, of how this would eventually fail, is when that poly-A tail starts to degrade. And I'll replay that once we talk about it. So the poly-A tail is protective, but mRNAs don't stick around forever. They have a finite lifetime. And the first thing that starts to happen as mRNA degrades is deadenylation. So the A bases at the 3' end fall off. And what that results in is that the PABPs also fall off because their binding motif is gone. This allows the decapping enzyme to then come in and take off the 5' end, as shown here, And now with both ends free, exoribonucleases can chop up the entire transcript. So this confers onto RNA a limited lifetime and a half-life. Yeah. So that's one way, but why, can you think of another reason why you wouldn't want a transcript to be around forever? Yeah. Exactly. So if you don't want to just keep making the same protein again and again, because if a transcript were to last forever, that cell is stuck making that protein for the rest of its life. But the lifetime of mRNA allows another chance for the cell to be in different changes and respond to its environment. So that's another form of regulation where we won't talk about this in detail, but other modifications can modulate the lifetime of mRNAs. And that's another way that a cell can decide if it's done making one protein or it needs to start making another. Any other questions? All right, so now that we've talked about that 5' and 3' modification, which occurs first, we're now going to talk about splicing. And to remind everyone what splicing is, it's taking out the introns, which are considered to be non-coding parts of DNA, and putting all the exons together. So you might remember a long time ago when we talked about protein structure, we talked about different parts of the protein having different functional capacities called domains. So proteins had catalytic domains, enzymatic domains, binding domains. And the way this relates to the DNA is that exons that are next to each other, or pairs of exons, tend to code for the same domain. So this adds modularity in how proteins are translated. So again, you have transcription, you have these exons separated by introns, and then we need to get the introns out of the way to make this mature mRNA. We're going to go back to what Emily referred to earlier about the naming conventions and the numbering conventions for mRNA. So the mature mRNA is shown on the bottom, It's just the exonic sequences, and the numbers correspond to the order in which the transcription happened, and also in the order that the translation will happen. But if you go back pre-splicing, you'll notice that the introns aren't numbered. The numbers skip over the introns and continue from the exons. And this is done by convention, just to know when you're mapping proteins back to the mRNA, you can focus in on just the coding parts of the mRNA. And then one last feature I'll point out here is that exons are multiples of 3. And do you guys know why you would need them to be multiples of 3 nucleotides? Yeah, but you're right, yeah. So one amino acid codon is made up of 3 base pairs. So again, it adds modularity. If you stopped a codon in the middle of an exon and it didn't get spliced with the perfectly adjoining codon, that protein wouldn't get translated. Yeah. Yeah, it's more important in the exon because that's what gets made into the protein. Yeah, did you have? Oh, sorry. Okay, so how does splicing occur? Splicing requires the, again, assembly of something called the spliceosome. And it's a large macromolecular assembly of many different proteins and this other type of RNA that I'll just get to in a minute. And I want to say right now, like, splicing is something that's still not very well understood about all the functions it creates and how it exactly happens. And it's an active area of research, but we're going to give you a picture of like what is known. So where does splicing occur? It occurs as splice sites, which are usually at the junctions of exons and introns. And one signal that's known for splicing are these 2 base sequences on either end of the intron. So at the 5' end of the intron, which would be over here for this one, you have a GU sequence. And on the 3' end of the intron, which would be over here, you have an AG sequence. if you think about just statistically the billions of base pairs that are in the genome, these two base pairings would happen with like a lot of frequency, yet splicing doesn't occur at all of the occurrences of these base pairs, which also leads to us to the fact that there's other specific signals that we don't yet know about for splicing. So these two we know and you should know. So again, the splicing reaction is catalyzed by a structure called the spliceosome, and in it are proteins and these specific types of RNA called small nuclear ribonucleic particles, or SNRPs, which is kind of fun to say. So we won't get into the nomenclature of every single SNRP, but we're going to talk about two that are particularly important for the splicing reaction, and and the flow of the order in which this reaction occurs. So again, this first strand here is the pre-mRNA, so it has 2 exons with the intron in the middle. At the 5' end of this intron is that GU sequence we just talked about, and on the 3' end is the AG sequence we just talked about. In addition, there is this other sequence shown here as an A base called the branch point sequence, and this is also critically important in the splicing reaction. So what happens first is that a U1 SNRP, so this is a SNRP, it's a RNA entity, binds the GU sequence. Then a U2 SNRP comes in and binds the branch point sequence. And then these two SNRPs can come together to bring those two parts of the intron close to each other. I'll point out that this cartoon here is wrong. This exon should go all the way to the GU there. So what happens then is kind of crazy. This A base attacks the G base in that first 2 base sequence. And this leads to the formation of this chemical feature between the A and G bases, and this first exon gets cut out. This loop is called a lariat, I guess as an homage to cowboys, and you still have the other exon bound to the other side. But then this free end of the first exon attacks the second consensus 2 base sequence on the other end of the intron. And these are transesterification reactions. And once that second attack happens, you get these 2 exons released as bound to each other and the lariat on its own. And that lariat gets rapidly degraded. So any questions on that? That's a lot of different steps of things. Yeah. What is it? Like, what's the sequence or what's its purpose? Yeah, so it's also, it's a A base as shown here, and it's also conserved and is required for this splicing reaction. Yeah. I think as far as we know right now is just the A. But it's kind of crazy. Like, again, statistically speaking, there's no way that's the only thing that's required for this. All right, so that's everything on splicing. We can go into a little bit of an intro on what regulation of these processes look like. That we'll get into more detail next week.
Okay, so can you guys hear me? All good? Okay. So next week, Dr. James is going to give the full lecture on transcriptional regulation and circle back fully to MODY and anabolic steroids. I'll just give you a little bit more background. So when Dr. Janes gives his lecture, he's going to pretty much interchangeably use the words gene expression and regulation of transcription, because basically if you express a gene, that's the same thing as you want it to be transcribed to produce the protein. So they kind of go hand in hand. So MODY and anabolic steroids both act via these insulin gene regulation pathways. And so anabolic steroids, there are many, many adverse side effects to them, but why would people use them? So the main two ways people use them are doctors prescribe them as a therapeutic agent, and the more common way that I guess most of you probably have seen publicized are athletes use them. So athletes will use them because you can get increased muscle mass and increased strength as a result, and decreased recovery time with your workouts. And the therapeutic uses by doctors can treat some different diseases that you can see here.